Today we're going to be looking at a slightly different type of cell division, something called meiosis, and its role in sexual reproduction. But I first want to go back to really a topic from last time. I want to go back and quickly review mitosis, because some of the same mechanics are at work in both mitosis and meiosis, and certainly a lot of the same terminology is at work in both mitosis and meiosis. So again, just a quick reminder on mitosis then. You may recall then that we typically are going to start with a diploid cell, which means that there are two sets of chromosomes, and in humans we have two sets of 23 chromosomes. And in interphase, those will replicate to form 4 sets of sister chromatids, and then those will eventually be separated into 2 cells, each with 2 sets of chromosomes again. So at the end of the process, then we have 2 genetically identical cells. And again, it's a little bit different than the way your book or most textbooks present this, but again, this is the way that I'm comfortable doing it, so hopefully you'll get comfortable with that as well. All right, so I'm going to go to the overhead camera again and kind of work through that process of mitosis and make a couple of comments about it then. So again, we would be starting with the diploid cell. So what we're looking at here, these Play-Doh models represent one set of 23 chromosomes you inherit from dad, one set of 23 chromosomes you inherit from mom, so we have a 2N cell. And what would happen in the S subphase of interphase is that the chromosomes will be replicated and attached to one another by a centromere. And at this point, we refer to these as sister chromatids. Then the cell would enter prophase, and in prophase, you may recall that what happens is that the chromosomes, sister chromatids begin to coil and condense, become visible. Nuclear envelope will disintegrate. Centrioles or centrosomes will move to either end of the cell and produce a mitotic spindle between them. And so let's go ahead to that phase and look better look at prophase. So again, a mitotic spindle will form. And then the spindle fibers will connect to the centromeres and position the sister chromatids at the equator of the spindle, and we refer to that as metaphase when they're positioned in the center. And then also, again, we would have centrosomes at either end, astral fibers radiating out from that. And I should mention that only animal cells will have the centrosomes and astral rays. Plant cells do not have centrosomes or astral rays, but plants also do have a mitotic spindle. So these are required in animal cells, but not required in plant cells. And then in anaphase, the centromere will replicate, and what were sister chromatids now will be chromosomes and pulled to either end. And then telophase would involve cytokinesis, dividing the cell in 2, and then we would end up with 2 cells, each of which have their 2 sets of chromosomes, so we're back to the diploid condition. Now, one thing I do want to mention, because this will play a little bit of importance when we talk about meiosis, is that when we're at metaphase, it doesn't really matter how these line up. And also I should mention that the homologous chromosomes and chromatids don't really have anything to do with one another in mitosis. So they can line up in any position at metaphase. We can flip them, we can do anything we want, but at the end of the process, when they separate, they're still going to be genetically identical. So this is something I'll come back to as we discuss meiosis. All right, so let's consider this other type of cell division, meiosis. Okay, so meiosis then is a different type of cell division. In meiosis, it's referred to as a reduction division. What it's going to do is cut the number of chromosomes in half effectively, reduce ploidy by half. So again, it will involve 1 round of chromosomal replication, but this time 2 rounds of cellular division. So again, we'll start off with 2 sets of chromosomes. Those will replicate to form 4 sets of sister chromatids, which will be divided into 2 groups, and then those will be divided again. So we have 2 rounds of cellular division. So at the end of the process, instead of with 2 cells, we'll have 4 cells that instead of being diploid will also be haploid. So again, we start with diploid cells, end up with 4 haploid cells. And what's also important to understand about this process is that these 4 haploid cells are genetically dissimilar to one another, and they're obviously genetically dissimilar to the cell we started with because they're half the number of chromosomes, but they're also genetically dissimilar to one another because of 2 processes that we'll discuss in a few minutes, one being crossing over and the other being independent assortment. So we'll come back to those. I'll talk about those several times in a few minutes as we look at the process. Now, why reduce the number of chromosomes? Well, this plays a role in sexual reproduction. So sexual reproduction, of course, takes two. And if you imagine sexual reproduction and where sexual reproduction, or what we need sexual reproduction for, it's to produce new individuals integrating genetic material from two different individuals. So the bottom line of this is that it will increase genetic diversity. So that's really the importance of sexual reproduction, and it's a theme I'll come back to several times. So whereas mitosis occurs throughout our body, every cell in our body is potentially capable of mitosis. Meiosis only occurs within the gonads, only within the ovaries of a female and the testes of a male. And again, the point is to cut the ploidy in half, because if you think about what would happen if you had a sperm that's diploid and an egg that's diploid, and same as our cells, and if those fuse together, then the next generation would have 4 sets of chromosomes. And then if they produced gametes that had 4 sets of chromosomes and 4 sets of chromosomes, the next generation would have 8 sets of chromosomes. And if they each produced gametes of the same ploidy, you'd have 16, and so on and so on. So the number of chromosomes would accumulate, double in each generation, and before long there wouldn't be room for all the chromosomes in a cell. There's barely enough room as it is. So what meiosis does then is that we inherit 2 sets of chromosomes, and we get to pass on 1 set of chromosomes. So we get 2 sets of 23, one from each parent, 1 set of 23 from mom, 1 set of 23 from dad. We get to pass on 1 set of 23 to the next generation. Then when our partner contributes their set of 23, we're back to a diploid condition in the next generation, so that each generation then is diploid because our sex cells have half the number of chromosomes. So again, if we look at this cycle then, of course, males and females, males will have testes, females have ovaries. Meiosis will occur there. And again, those haploid cells that we end up with in meiosis then become the sex cells, either sperm or ova. And kind of a generic term for sex cells is to call them gametes. And the sperm, of course, will swim and fertilize the egg, and another term for fertilization is syngamy, which means fusion of the gametes. And then that will generate a diploid cell called a zygote. And then that zygote will mitotically divide to generate the new organism. So again, meiosis is required for sexual reproduction to produce the gametes, the haploid cells. And then once they fuse together and we're back to the diploid condition, mitotic division will create the trillions of cells of which we are composed. All right, so let's go ahead then and look at some of the mechanics of meiosis and discuss some of the peculiarities of meiosis. Oh, and again, bottom line, generate genetic diversity. So again, if you look at these 2 girls, my daughters, they have the same parents, yet they, at least in my mind, they look quite a bit alike, and they have very different personalities and so on. So that's the bottom line with sexual reproduction. Its benefit is to increase diversity of the species. And it seems to be important because virtually all organisms have a sexual cycle or sexual reproduction. All right, so as I mentioned before, meiosis is going to use a lot of the same terminology that we used in mitosis, although the outcome is a little different at the end. So again, we're going to end up with 4 haploid cells that become the gametes. So again, there's basically 2 rounds of division, meiosis 1 and meiosis 2. And in meiosis 1, we're going to have a phase, interphase 1. And you notice there's an asterisk by that. And it's an important phase because that's the phase where chromosomes replicate. So chromosomes are replicated to form the sister chromatids. Then prophase 1 is an important phase because this is where crossing over occurs, and we'll talk about that in a minute. That proceeds to metaphase 1. Metaphase 1 also has an asterisk because this is where something called independent assortment occurs. So again, I'm going to emphasize crossing over and independent assortment. So, keep those in the back of your mind right now. Then we'll have an anaphase 1 and a telophase 1, which results in 2 cells. Then there's a second round of cell division, which we call meiosis 2, which will have or may have, it depends on the type of cell, a short resting phase and interphase 2, followed by a prophase 2, followed by a metaphase 2. And you'll notice that that has an asterisk because that's also, again, where we're going to have independent assortment. Now, I want to also emphasize here that interphase 2, there will not be replication of chromosomes. And in prophase 2, there will not be crossing over. So I haven't put an asterisk there. And again, followed by anaphase 2, and then telophase 2, where each of those 2 cells divides, and we end up then with 4 haploid cells. All right, so again, where we're headed first, we're going to look at meiosis 1, which would be the first round of division. So this diagram doesn't really show us an interphase 1, but I'm going to emphasize an interphase 1 because that's where chromosomes replicate, followed by a prophase 1, where nuclear envelope disintegrates and crossing over is going to occur, metaphase 1, where our chromatids are going to line up at the equator, anaphase 1, where they'll be pulled apart, and then telophase 1, where we get cell division. So let's go ahead and look at this with the overhead camera and kind of work our way through some of these important aspects of meiosis. So again, we're going to start off with a diploid cell. And again, this will represent one set of 23 from one parent. We'll say this is from dad, this is from mom. And in interphase 1, these will replicate and form sister chromatids being attached by the centromere. And then as we go into prophase 1, the spindle will form, and this time we call it a meiotic spindle instead of a mitotic spindle. But what's important about prophase 1 is that this is where a really an important process occurs called crossing over. Now you may recall in mitosis, the homologous chromosomes really don't have anything to do with one another and they'll line up at the equator independently of one another. But in prophase 1 of meiosis, what happens is that the homologous chromosomes do associate. There are proteins that will line them up gene for gene. And so the 4 sister chromatids of a particular type of chromosome then will form a group of 4 chromatids. And we call this a tetrad, group of 4, a tetrad. And there are additional enzymes then that will cut and splice these chromatids. And again, typically what will happen is that they may cut and then recombine with themselves. But occasionally what will happen is that one sister chromatid from one parent will be cut and spliced with a sister chromatid of a different sister chromatid. When that occurs, we call that a crossover. And that's a significant event because what this does now is that we've inherited chromosomes of one type, but this is generating chromosomes of a completely new type. And I'll come back and emphasize that in a second. So crossing over occurs only in prophase 1. Then the entire tetrad will be positioned at the equator in metaphase 1. Now, at metaphase 1 and metaphase 2, we have something called independent assortment that occurs here. And so I'm going to introduce another tetrad. So let's say these are the tetrads for chromosome 1. These are the tetrads for, let's say, chromosome number 6. What is meant by the term independent assortment is that this group of chromatids, this tetrad, will not have anything to do with an additional tetrad at metaphase. So how this group lines up has nothing to do with how this group lines up. So we could invert this. This one has nothing to do with that one. They can be in different positions here. And this is significant because depending how these line up at the equator, it will give us different genetic outcomes because the tetrads, because of crossing over, or the sister chromatids, I should say, are no longer genetically identical. They're no longer the same because of crossing over. We have new combinations here. So the fact that one tetrad or group of chromatids of a particular type have nothing to do with another group, we refer to that as independent assortment. And so what that means then is that our chromosomes are randomly distributed in the gametes at the end of the process. Okay, so following our sister chromatids then, in anaphase 1, the tetrads will be separated. And so again, now we have sister chromatids that are no longer identical because of crossing over, and those will be pulled to either end. And then in telophase 1, the cell will pinch in two, or if it's a plant cell, it'll build a cell wall in between, and we will end up with 2 quote-unquote daughter cells, although this is not the end of the process at this point. Okay. I also now will, I think I'll go to an animation and very quickly show you the events of meiosis 1 in this animation. And again, I'll kind of talk over that as we go along here. So anyway, don't get too excited here the way it starts out. We'll get to the biology in a second. But again, meiosis is going to occur within the gonads, within the sex cells. And so they're just calling this interphase, but this would really be interphase 1. And so again, we would be looking at the nucleus. And what's going to happen here is that we're going to have replication of the chromosomes. It's getting there. So we're now looking at the chromosomes in a little more detail. They will begin to replicate. And while it's not emphasized here, they'll be attached by centromeres, and so we would refer to these as sister chromatids then. And then they begin to coil and condense. Initially, that's not visible, but when that does become visible, we would then be in prophase 1. And so again, prophase 1 would be as any other prophase we've talked about. So we're going to see production of the spindle, in this case the meiotic spindle. Chromatids continue to coil and condense, and they'll become visible to us with the light microscope. And then the tetrads will associate. So again, a very important event. The tetrads are going to associate, and crossing over will occur between different types of sister chromatids. Meiotic spindle continues to form, nuclear envelope disintegrates, and the group of 4 chromatids, the tetrads will be assembled at the equator of the spindle. So this would be metaphase 1, which we're headed towards. So once they're all at the equator. And so again, there would be 23 groups of tetrads then at the equator of the spindle. And then the sister chromatids of the tetrad will separate. And this is kind of a dangerous time in this process because a lot of breakages can occur here. This is actually a process that's fraught with hazard. So sexual reproduction has a downside that we'll come back to in a second. And then, of course, telophase 1 and cytokinesis, chromatids have been pulled to either end, and then the cells go ahead and separate. All right. So at the end of this process then, really what we're looking at is that each cell has 2 sets of chromosomes, but they're attached in the form of sister chromatids. So it's a 2-in condition, but kind of a weird 2-in condition. And now then we would enter meiosis 2. And so again, we've already talked about prophase and metaphase 1. I'm going to have to scroll forward here a little bit. And I want to quickly come back to crossing over, again, to talk about what happens here. So in crossing over, again, we'll have replication of chromosomes in interphase 1. The 4 will associate in a tetrad. Enzymes will cut and splice so that it's potentially crossing over will occur. So sometimes one chromatid will splice with another type of chromatid. And when we look at the downstream effects here, when we eventually separate these into individual chromosomes, what's going to happen is that we may have some chromosomes that do not have any crossovers, but we will have some then that are completely new types of chromosomes. So whereas you may have inherited a chromosome from dad that has, where you have, it's coding for, say blue hair and blue eyes, and mom has red hair and red eyes, you now on a single chromosome could have blue hair and red eyes, or red eyes and blue hair. So again, crossing over won't create new traits, but it creates new combinations of traits. And so that's an important thing to understand. Okay, and so again, downstream effect then would be that all one set of 23 that you're passing on isn't just what you've inherited from mom or dad. They're new chromosomes. They're combinations of your mom's and dad's chromosomes that you are passing on. Okay, now I'm going to scroll ahead here a little bit to look then at meiosis 2. So in meiosis 2 then, it's much more like mitosis. We're basically just going to finish out each of the 2 cells that are the result of meiosis 1 are going to go through another round of division. There's not going to be replication of chromosomes. There's not going to be crossing over. And each of those 2 cells forms 2 more cells, which will then be haploid cells. And again, looking at it in a little more detail then and looking ahead where we're headed, so we will have a prophase 2, in which there will be no crossing over, metaphase 2, in which the sister chromatids line up at the equator, and independent assortment will occur here. In other words, different sister chromatids will not associate with one another, and how they line up does not affect how another sister chromatid pair lines up. And then those will be separated in anaphase 2, and then the cells divided in telophase 2. And at the end of telophase 2, then we have 4 haploid cells that are just genetically dissimilar because of crossing over and independent assortment. So again, I'll show you this in the overhead again, but in prophase 2 and metaphase 2, again, pretty typical, very much more like mitosis, and then anaphase 2 and telophase 2. So let's go ahead and look at this again from an overhead perspective. And I'm going to go ahead and kind of eliminate our spindles. We'll just go ahead and say, look at these now and say, okay, these are in different cells now, 2 different cells. And so what would happen is that a spindle would form in each of those 2 cells. And again, the cell may enter an interphase. Sometimes the cells will kind of rest a little bit, but they normally go directly into a prophase 2. And so in the prophase 2, if a nuclear envelope is formed, it will dissolve. New spindles will form, new meiotic spindles will form in each cell. And then in metaphase 2, the sister chromatids will line up at the equator. And again, how one sister chromatid lines up has nothing to do with how another sister chromatid lines up. And so this is significant. So I just want to focus on this one here. So as we go to an anaphase 2 then, these can separate, and these will separate. And this would give you, and eventually pinch off in telophase 2. So this would give you one genetic outcome compared to this. But again, let's go back to where we were. If we turned this one over, this would give us a different kind of genetic outcome. So the fact that different tetrads and sister chromatids at metaphase 1 and 2 function independently means that we have the potential to have all sorts of genetic outcomes here, and almost an infinite number of genetic outcomes. And that's why it's virtually impossible then to produce 2 gametes that are the same. So again, in each cell you would have chromatids lining up at the equator in metaphase 2. They would be separated in anaphase 2. And then each of these would form an individual cell. And so at the end of the process, you would have 4 cells, each with one set of 23 chromosomes, and they would be genetically dissimilar because of crossing over and independent assortment. So again, kind of looking at the overall process, again, as we've talked about, we go from 2 N cells eventually to 4 haploid cells that are genetically dissimilar, and the function of those cells will be to serve as gametes because of crossing over and independent assortment. Now I want to come back briefly to independent assortment. And the point here is, and at the end of the process, we inherit 2 sets of chromosomes. And so we have some options here. We potentially could pass on a set of dad's chromosomes or a set of mom's chromosomes. Or we could scramble those sets and pass on some of dad's chromosomes and some of mom's chromosomes. And so that's what independent assortment accomplishes. So the analogy I use here is, let's say you have a trunk of clothes that you inherited from your mom and a trunk of clothes you inherited from your dad. And let's say you have a child and you decide you're going to give your child a trunk of clothes. You could give them a trunk of what you inherited from your dad. You could give them a trunk of what you inherited from your mom. Or you could take some clothes from one trunk and some clothes from the other and come up with a new trunk that has some of mom and dad's clothes in it. And that's what independent assortment does. Now, when you compound that with crossing over, so what crossing over would be is that you're not only going to pass on some of dad's clothes and some of mom's clothes, but what you would then decide to do is, oh, I'm going to cut a shirt of mom's and a shirt from dad's and cut those apart and sew those together. And so I'll pass on a shirt that's half mom and half dad's. That's what crossing over does. So we not only don't pass on a complete set of chromosomes that we've inherited, we don't even pass on individual chromosomes that we've inherited. We cut and splice those together to pass on completely novel proteins so that we're passing on combinations of traits that are unique. Now, meiosis has a lot of problems with it. The tetrads, the chromatids getting all tangled up can create all kinds of problems. Lots of breakages occur here. So one of these, for example, is a translocation. So one of the things that can happen is that a chromosome can break, and then during meiosis, it'll get spliced onto a completely different chromosome. So maybe a portion of chromosome 1 gets spliced onto chromosome number 4. We refer to that as a translocation, and it may get stuck on the end of the chromosome. Another possibility is that that broken chromosome may splice itself into the middle of another chromosome, and again, that potentially is problematic. So for that particular individual, this isn't necessarily a problem because we haven't really lost any genes. It's just that the genes now are in a different chromosome. With some genes that will be problematic, and other genes it wouldn't be. But when you make your gametes for the next generation, it could cause problems there. So it might not affect you personally, but it could affect the next generation. Now, another kind of mistake that occurs, or chromosomal abnormality, which occurs during this process, are inversions. And in inversions, again, during prophase 1, during crossing over, what can happen is that as cuts and splices are made, is that a section of a chromosome actually will get inverted and then spliced back into the same chromosome. So again, you haven't really lost any genes here, but what you have done is change the order. so that if this occurs in your gametes then, and you're trying to cross over in the gametes, it could create problems in the gametes that you produce, even though it may not affect your health personally. And then also, again, there are a number of so-called non-disjunction events which can occur. For example, if we go back to metaphase 2, for example, and let's say we have the equator here, and let's say the spindle is going this way, and these are going to be pulled apart, we should be able to separate the two sister chromatids, and they should be pulled to either end. But sometimes what can happen is that the centromere won't replicate, and perhaps both chromatids go to one end and don't go to the other end of the cell. So eventually what we're going to have is a gamete that lacks a particular chromosome and another gamete that has twice as many chromosomes that it has. So this non-disjunction means that the centromere doesn't replicate properly, and we don't get proper separation of chromosomes, so that we have some chromosomes which are doubled up in a gamete and other gametes that will have omissions. And of course then when they join up with another gamete, you'll either have too many chromosomes or too few chromosomes. And so again, when we look at karyotypes, this would be a normal karyotype where we have 2 chromosomes of each type in an individual. And again, with Down syndrome, trisomy 21, because one of the gametes was carrying an extra chromosome. So again, as we look at sexual reproduction and the role of meiosis then, is that meiosis really is hazardous. It's really problematic because there are a lot of mistakes that occur here, and we produce a lot of abnormal gametes. So what's the benefit? Well, the benefit, as I've mentioned before, apparently is diversity, genetic diversity in a population. That's really what meiosis and sexual reproduction are all about. And if you think about that, if you think about asexual reproduction compared to sexual reproduction, if we were all genetically identical, let's say a new disease appears, if I die from that disease, you're going to die from that disease because we're genetically the same. But if you have a better immune system for me, maybe that kills me, but maybe you survive because of the diversity in the population. So again, asexual reproduction in a lot of ways is more convenient. You don't need to mate, but you have a genetically homogeneous population. Sexual reproduction is much less convenient, but the genetic diversity helps preserve the species.